All right, howdy, Ags. We're back for a short installment of skeletal muscle mechanics. So we're going to talk about muscle cross-sectional area size. We're going to talk about muscle length. Start to get into some of those mechanisms and how we can increase active force. And we're really trying to give it some force and power. So we're going to start. We finished up with excitation, contraction, coupling last time. And today, or in this section of today's lecture, we're going to talk about, first of all, muscle size and cross-sectional area, muscle length, and then we may get into something called ray coding, how we control the amount of force, not too much, not too little, get it just right. So skeletal muscles actually have a limit in terms of how much force they can produce. So I'm going to draw your attention down here to a graph. There are a number of lines on it, but I'm going to draw your attention to this top one. This is a healthy mouse, by the way. And this is the extensor digitorum longus. So it's a muscle that has mostly fast twitch fibers. Mice, as you know, mostly, or you may not know, have mostly fast twitch fibers. They're small, quick, little creatures with lots of fast twitch fibers. They can get around. They're not big enough to produce a lot of power, but based on their relative size, they can certainly produce enough power to burrow into the ground and into their den, etc. So you can see a max here of about 90 newtons per centimeter squared. That's kind of the max we see. In fact, I'm usually skeptical when I see data that's very close to the max, because we usually don't see it. Most muscles can't produce quite the max, but this is kind of the ceiling, because we can only stuff so many cross bridges into one muscle. We can only stuff so much actin and myosin in. So the only way we can actually increase force for an individual muscle fiber is to increase its cross-sectional area or size. So when we want to increase force production in a muscle, and strength training will do this, there are two mechanisms. One is indeed increasing fiber size, cross-sectional area in mammals. In birds, they can have something called hyperplasia instead of hypertrophy. That's an increase in the number of fibers. For a creature that has to fly a lot and might have to migrate a long way, thousands of miles, that might be a good idea. But we don't really think it happens very much in mammals at all. Sometimes when there's a lot of muscle damage, it can appear that there are more fibers from time to time, but they usually get weeded out. So in the end, you don't really have a significant difference. That often happens with things like muscular dystrophy. There was some confusion in the 1990s because doing histology in skeletal muscle is really challenging. The muscle is frozen. That's the best way to be able to basically slice it, if you will. And when you put it on slides, et cetera, and take a look at it, you have to make sure that the fibers aren't split. So there's some specific ways that we try to keep these muscle fibers from getting what we call freeze damage. That's what happens to basically meat, like chicken or beef or fish or whatever else you stored in the freezer. Maybe it's stayed there a long time. We had this ham that was in our freezer for about a year and a half, and we had to get rid of it, partly to make room for all of the frozen supplies and food that we've been storing up like everybody has. But it had a lot of freeze damage on it. And that's caused by thawing and refreezing and thawing again. And this happens especially when we have a muscle, or in the case of meat, something that has a lot of water. So if you think you want to go Michael Jackson, allegedly, and get your body frozen in liquid nitrogen to preserve yourself, I got bad news for you. It's not really going to work, because you'll get a lot of freeze damage in tissues because they have water in them, and they're going to form ice crystals, which will permanently damage any cells that might have been alive. So sorry to bust that myth for you. Supposedly, maybe Michael Jackson got himself cooled or something, so they can fix him later. Anyway, so because muscles have this limit in terms of how much force they can produce, we either have to make the muscle bigger, or we can recruit more muscle fibers by increasing the efficiency of the recruitment of motor units, which again are the alpha motor neurons and the muscle fibers that they innervate. Okay, so how do we do that? Well, with resistor training, the idea is to put enough stress on the muscle that the muscle has to produce a lot of protein synthesis. And it has to be done over time. So just weightlifting once every 6 weeks is just going to make you sore. It's unfortunately not going to cause hypertrophy. The muscle soreness can actually jumpstart the process, but you have to keep doing resistor training and adding in that mechanical stress. So it'll take 2 to 3 days a week. And by the way, let me see if I can write on here. No, I can't. Maybe I can. There we go. It's a little hard when I have it as a separate window to try to manipulate it. But this is the percent of your 1 rep max. In other words, your maximal lift. This is the intensity that you need to be able to produce with a workout enough mechanical stress that the muscle will start to produce a lot more protein synthesis. Protein degradation goes up. The protein synthesis goes way up. So in time, what you're going to start to do is accumulate muscle mass. And that's going to happen by adding on microfibrils in the muscle fiber. And that's where the myosin and actin are. So you can add a bunch of those. The muscle gets larger. We think that some of the satellite cells will actually add in a few more myonuclei to help with regulating the cell. So the more DNA a muscle fiber has, the more protein it can produce. And so because muscles are long, they're multinucleated. They have lots of nuclei. And again, they're right on that cell membrane. So this is a really interesting graph. I copied it over from a paper where they had men and women, young men and women in a particular study. This was upper body strength, elbow flexor muscle strength on the y-axis. The x-axis is the cross-sectional area of the muscle itself. So I think they measured it through MRI, if I remember correctly. And you have men and women. Remember, this is the upper body. If this was the lower body, there would be a lot more overlap between men and women because the difference in muscle mass for like the quadriceps, hamstrings, gluteal muscles, etc., it's not that much different. So one of the things to notice is that a lot of the data points are very close to this line. If you plotted its or calculated its correlation or did regression analysis, you would find a pretty tight fit overall. So there isn't really a difference between men and women besides basically the cross-sectional area of the muscles, which we see a little bit more again with the upper body than the lower body. But a lot of this depended on fitness and genetics. So you see some women and men here on this line overlapping with each other. So if we want to increase force production, we'll increase the number of motor units we recruit and increase the cross-sectional area of the muscle fibers. This takes a little longer. The neuromuscular adaptations happen in the first few weeks. We start to see significant hypertrophy about 3 to 4 weeks out. Takes a few weeks before your body thinks you're really serious about the workout because it's expensive to increase muscle mass. So changing muscle length, stretching a muscle, not only increases passive tension that we talked about a while ago, like stretching a rubber band, but it also increases active tension. And the mechanism is it puts the actin and myosin strands, the heavy chain and light chain molecules, or the thick filament and thin filament, it puts them in an optimal position where you can get the greatest number of cross bridges between actin and myosin. If the muscle gets too short, as we've demonstrated before, you lose a lot of force. If you stretch those muscles, it actually increases the amount of force that they can produce, whether it's finger flexor muscles or your knee extensors or plantar flexors. By stretching them, you get a number of benefits. So let's talk about some of the mechanisms. How does this happen? How do we increase muscle force with an increase in muscle length? Well, again, some of it has to do with optimizing the number of cross bridges between actin and myosin. Some of it is initiating the stretch reflex, and that is where muscle spindles pick up the stretch. The more rapid the stretch, the greater the effect on the stretch reflex. The stretch reflex will then, through interneurons that are in the spinal cord. So I think this is a simple reflex, and then there's a single interneuron between each alpha motor, between each afferent nerve and each alpha motor neuron. So the idea is that with a stretch reflex, when a muscle is stretched rapidly, the muscle spindles react, send that information to the spinal cord. The spinal cord then relays the information to alpha motor neurons, which are going to increase the amount of force generated. And that happens really fast. These are really fast reflexes. So the stretch reflex and something called the extensor thrust reflex, these are some of the fastest in your body that are related to walking, running, throwing objects, etc. So the stretch reflex can really help. If you, we don't want to use the term bend down, if you flex your knees, hips, plantar flex the, or dorsiflex the ankle before you jump, that will initiate that stretch reflex. It will put those muscles in a position where they have more cross bridges. Also, when you stretch a muscle, it increases the amount of calcium that's released from the sarcoplasmic reticulum. We can take advantage of this, especially when we're, maybe you're going to hit a tennis ball, we go for a windup. That fast windup is going to maximize passive tension and active tension through these mechanisms. So this is kind of what we want with active tension. When we get near the end of the range of motion, we got a peak that's close to our resting length, and about 120% of the resting length, that's about where we see the end of the range of motion. If we add in passive tension, then we get a pretty good peak right in here. So that's where you can produce the greatest amount of force. So this is kind of an old classic curve from physiology and exercise physiology classes. So even at the sarcomere level, that's what's highlighted here with 1.2 microns, 1.6 microns. People actually measure these at the sarcomere level. So at the end of the range of motion, you're about right here. This is kind of between 100 and 120% of resting length. This is that Goldilocks zone where we can maximize the number of cross bridges available. When the muscle shortens, they start to crowd together, and some of the binding sites at actin either get covered up or they're simply not available. If a muscle stretches too far, then we get a significant reduction in the amount of force that the muscle can produce, like hyperextending the wrist, for example. But usually we'll reach the end of the range of motion significantly before that. So optimal length is key in terms of stretching. We've done this exercise before, right? So again, looks like this person cheated too. So if you take that pen or pencil and you flex your wrist and try to grab it, you can't produce a lot of force. Extend the wrist and those finger flexor muscles are now stretched, and you can produce lots of force. So big difference there. And again, 120% of resting length, that's kind of ideal. Something interesting, muscles with a high percentage of fast twitch fibers reach peak tension at greater lengths than slow twitch. So what the heck does that mean? So that means if someone kind of scrunches or squats down before maybe a volleyball spike or for a jump shot in basketball, or maybe before they're going to jump up, maybe it's a shortstop going to jump up to try to catch a line drive. If they have a higher percentage of fast twitch fibers, the more stretch they produce, the greater the amount of force they actually get out of it. So fast twitch, a person with a high percentage of fast twitch fibers gets more benefit from a wind up or a stretch, etc. When we get older, we tend to lose a significant percentage of fast twitch fibers. We also have, and this is partially related to that, we also decrease the length at which peak tension occurs. So when you get older, you don't get quite as much of a benefit from a wind up or a quick stretch before you produce some kind of task. So again, these classic curves, you have to add together or get the sum of active tension plus passive tension. Sum those suckers together. So this is a reminder, where you guys skipped the slide very quickly. So again, muscle size, the greater the cross-sectional area, the greater the number of cross bridges, the greater the amount of force that can be produced. For a muscle of any given size, if we stretch it, we increase the amount of force that the muscle produces, and that's through stretch reflex, optimizing the number of cross bridges, increase calcium, etc. All of those mechanisms increase the amount of force that the muscle can produce. The next thing we're going to look at and talk about today is rate coding or frequency modulation. FM, like your FM radio. That's the same way we code information on your television or on your computer. It's digital information. It's binary. It's 1 or a 0. And motor unit recruitment. So these factors can help us control the amount of force. If you lift something like a pen, you don't want to produce the same amount of force that you do when you're lifting, pretend this is like a 30-pound weight. So when you're lifting a heavy weight, you obviously want to produce more force. So how do we do that? We recruit more motor units and we increase the frequency of the action potentials coming down the alpha motor neurons. In motor neuroscience, they call it rate coding. In engineering, we call it FM, frequency modulation, as opposed to AM, which is amplitude modulation. That means if someone's playing a trumpet or guitar or another instrument, that let's say the louder the instrument is with AM, the signal on top of what we call a carrier wave would just be larger. But in FM, what we do is we code by frequency. The louder it is, the faster the frequency. That's exactly how your nerves work. So alpha motor neurons code information by frequency. You want to get the muscle to contract harder with more force, you increase the frequency of action potentials coming down those alpha motor neurons. So that's what the spine, the brain are doing. They're controlling the number of motor units that you're recruiting and the frequency of those action potentials coming down those motor neurons. So again, this is a reminder that hey, every single muscle fiber in a motor unit is basically like a clone of all of the other fibers. They have the same chemistry, the same histology. They're the same muscle fiber type. They're either fast twitch or slow twitch, and later on we'll divide fast twitch into 2A's and 2B's. There's the motor end plate, which is usually right in the middle of the muscle fiber. So when an action potential comes down, it causes these quanta. They're actually little vacuoles that form. Acetylcholine gets packaged in them, travels down to the synaptic cleft, goes across the synaptic cleft, binds to receptors, acetylcholine receptors, on the muscle fiber, and that causes sodium to rush in. That's what depolarizes the muscle cell. And the motor end plate, again, is usually in the middle of the muscle fiber. Once the action potential starts in the muscle fiber, it takes less than 2 milliseconds to cover the entire muscle. So it's almost, but not quite instantaneous in terms of depolarization. So again, we have fast twitch and slow twitch motor units. Here's the neuromuscular junction. This figure is almost correct. At least the muscle nuclei are near the membrane, so that's pretty good. It's close enough. Usually they're right on the membrane. And here we see the synaptic cleft. There's a lot of mitochondria. Glad they showed this. There's a lot of mitochondria near the cell membrane because there's a lot of ATP needed because you're moving ions like crazy back and forth. And a lot of times you'll need to pump them either towards the outside or towards the inside or into the mitochondria, etc. So with a motor unit, each axon splits up and contacts 20 to 2,000 muscle fibers. And again, muscles that have a small number of muscle fibers per motor unit, they're there for fine tuning. So again, ocular muscles, muscles that control finger movement, etc., where you need, in fact, your fingers, the dexterity in them is unbelievable. You can have these incredibly small deviations in pressure and movement, and you're able to feel this. This is why some people can be really good surgeons with a lot of practice. And in fact, they do really, really well when they have a microscope, when they have a camera that can blow up an image. So people can do really fine surgery on a knee or on an ankle, a shoulder, brain, etc., the heart, where you're working with really teeny tiny things. So like a coronary artery is not much in a human. It's not going to be much wider than a pencil lead. It's really pretty small. So it takes a lot of good fine tuning to do that. So that's the strategy when we need fine motor control. We just have a few muscle fibers per motor unit. Okay, so the whole idea is whether we're lifting a pencil or 50-pound bag or something in between, we want to be able to control the amount of force. So again, it's by frequency. FM modulation is good because it reduces noise. That's why we don't have too many radio stations on the AM dial. They're kind of cool at night because they can go a long way. But in terms of clarity and in terms of really good signal, FM's the only way to go because it reduces the amount of noise or errors. If you've ever done an ECG or an EKG, you probably noticed that you were not supposed to move or shake the leads. If you do accidentally, you'll look at the EKG and it'll have lots of squiggly lines. It might even look like they're in VFib or something, might scare someone. But the idea is that if we used amplitude modulation, every time we moved our arms, it would cause lots of noise. We couldn't figure out what was going on. Our coordination would be very poor. A much better way to do it is just like a computer does. It's binary, it's digital, we code by frequency. So as the frequency of action potentials increases, and as the recruitment of motor units increases, we can increase the amount of force that the muscle is producing. The mechanism is called summation. That summation is of calcium levels. The greater the frequency of action potentials, the more calcium that's in the cell, partly because we're limited by the amount of calcium we can pump back into the sarcoplasmic reticulum. So the faster the action potentials, the more that the calcium builds up and piles up inside the sarcoplasm. One of the things to remember, and this is why it's ray coding and why it's digital information, this is an action potential that you might see in a muscle fiber or in an alpha motor neuron. They all kind of look alike. So they're kind of a 1 or a 0 binary information. Most of the time, a muscle or a neuron is going to be somewhere between -70 to -90 millivolts in terms of membrane potential. And when there's an action potential, sodium rushes in, the cell depolarizes, you get up to about +20 millivolts, and then you come back down. There's usually a little bit of hyperpolarization and back to normal. So normal would be resting membrane potential. That's a 0. The action potential is a 1. So there aren't larger or smaller action potentials. That's just your bit, B-I-T, of information. So 1 or a 0. So a really good way to code information. And this is force on the y-axis, but it could be calcium. So when there's an individual twitch, there's an increase in calcium and therefore force. And then there's a calcium ATPase pump in the sarcoplasmic reticulum. It pumps calcium back into the SR, and then levels go back to normal, and so does the force. They are absolutely linked together. When we get an increase in frequency of action potentials, then before the calcium ATPase pump in the SR can get the calcium back, you see an increase in the amount of force due to, the increase in the amount of calcium that we see in the muscle. And that summation continues. An individual twitch, which is the force produced by a single action potential, is usually about 20% of what we see at tetanus. At tetanus, it simply means that even if we increase calcium more, we couldn't bind any more sites on troponin. That's what happens. Troponin, its binding sites for calcium, each molecule is 4 calcium binding sites, for whatever it's worth for you guys. They get full. And so that means that we've revealed as many actin binding sites for myosin as possible. So that's why you reach tetanus. You've reached the maximum number of cross bridges. You can't produce any more cross bridges. So you've maxed out. That's why you reach tetanus. Oops. Got a mind of its own here. Okay. Hold on a second. There we go. Let me slide things back into place. This is a real muscle, a real experiment. In fact, it's actually from one of the first experiments I did at Texas A&M. So we were actually fatiguing diaphragm muscles, believe it or not, trying to understand what makes them contract. We were looking at how reactive oxygen species like hydrogen peroxide, enzymes like NOx2, enzymes like xanthine oxidase, how they actually regulate muscle contraction, and what also happens in fatigue. Because oxidative stress seems to be blamed a lot for fatigue, and it does contribute to some fatigue, but it can also actually increase muscle force under the right conditions. So if you're a sprinter, for example, the last thing you probably want to do is take a lot of antioxidants, because it would actually reduce the amount of force you could produce during sprinting, maybe even during weightlifting. So that's going to sound a little bit weird. If you're overtraining, yeah, then you might want to take some antioxidant supplements, but you're just better off eating a lot of fruits and vegetables anyway, just doing it naturally. And your body will adapt to that a lot more easily than a supplement. So this was a fatiguing protocol. And what I want you guys to look at is this sigmoidal shaped curve, this treaded S-shaped curve. This is pre-fatigued, and then we have post-fatigue. So I wanted just to point out a few things. That as we increase the frequency of action potentials, and that's here on the x-axis, the muscle force or muscle tension goes up. And you can see that. The reason that you have that sigmoidal shape, you know, that looks familiar, doesn't it? Kind of looks like an oxyhemoglobin curve. Well, there's a reason that they look alike. Hemoglobin is made up basically of 4 subunits. Each one has iron. So it's like a clover leaf. There are 4 of these subunits together. If we just took one out and put it in a muscle cell, that's actually myoglobin. But as a tetramer in your blood, they're hemoglobin. They're red when they bind with oxygen. The reason you have this sigmoidal shaped curve is a property called cooperativity. And that means that for troponin, once a calcium binds to one of the binding sites, that alters and sometimes helps the ability of the next binding site to bind the calcium. This is also true for hemoglobin. So that's why you get this type of shaped curve. So we get a steep curve right in the middle. So at about 20, 30, 40 hertz or so in most muscles. This is a mixed muscle, meaning it's about 40% fast twitch and about 60% slow twitch. It's actually pretty close to 50-50 if I remember these particular muscles. So it's typical of what you might see in most human muscles, believe it or not. This was from a rat, by the way. Wasn't from a human. So as we're looking at this curve, you can see eventually it reaches tetanus. But in most ordinary things that we do, whether we're playing tennis or golf or swimming or running or walking, whatever you like to do, you're working mostly in this part of the curve. Okay, so what happens with fatigue? So we used a fatiguing protocol and demonstrated that the fatigue was in the muscles and the muscle cell itself. So even though, let's say we put 40 hertz of or frequency of action potentials down those alpha motor neurons. So we kind of simulated this in organ bath. And when we did that in the muscle that had been fatigued, you can see the amount of force or tension is dropped by almost a third. So significant fatigue, especially where we need it in the physiological range. The difference wasn't nearly quite as much with tetanus. So this is, and at the low frequencies is kind of normalized. So this is kind of a combination of mostly low frequency fatigue with a little bit of high frequency fatigue. This is the kind of thing you might see after a mile or a 2 mile or 5K race, where you'll be pretty tired for a while. The good news is that the fatigue you see with a 100-meter race or 200-meter race is the kind of thing that can, by walking it out and a little bit of exercise, getting that lactic acid out, if it's the 200, 400, or 800 meters, in about 20 minutes, you're probably ready to, you may not feel like it, but you technically might be ready to go for another round. So that's what happens with fatigue. It reduces the amount of force that's produced by the muscle at the same frequency. So there's a few mechanisms. Some of it happens at the contractile apparatus, where the sensitivity for calcium has been altered. Sometimes it is waste products, which include lactic acid, inorganic phosphate, some of the intermediates of glycolysis, and the Krebs cycle can also cause fatigue. So the number of mechanisms can actually lead to this. So I think it's getting stubborn here. That's actually about it for today. We're out of time and we're out of slides, so it worked out perfectly. So next time, what we're going to do is I'm going to see if I can go back to our, there we go, to our overview. We're going to talk about shortening and lengthening velocity, how we can alter the amount of force production and vice versa, alter the speed in which a muscle shortens or extends. What does that have to do with force? So we're gonna see another trade-off that we talked about. What are the effects of increased muscle temperature on the force production in skeletal muscle, particularly in vivo, because that's where we have most of the good data. When we do it in an isolated bath, you get actually some different results. Muscle fiber type, how does that affect muscle performance and mechanics? So in here, we're going to look at a number of curves that are going to be really important. Force velocity curves and power velocity curves. And then we're going to talk about reflexes. And I think we'll finish up next time talking about, Trixie's here saying, it's time, man, to just stop. Okay, so next time we're going to talk about some muscle injuries, delayed onset muscle soreness, the effects of aging, etc., and kind of wrap up the skeletal muscle section before we start to move on to bones, tendons, and ligaments in the next couple of weeks. Okay, that's it for this little episode of biomechanics. So I'm going to try my bumper music on the way out. You guys be safe. And we'll see you, well, probably tomorrow. Have a good one.